Hello friends, welcome to Meet the NEET. Today we are discussing the chapter Cell as a Unit of Life. Have you heard about endomembrane system? What is endomembrane system? As its name indicates, it consists of a membrane-bounded organelles which functions in close coordination with one another. They are endoplasmic reticulum, Golgi complex, lysosome, and vacuoles. Along with it, nuclear envelope is also considered as a part of endomembrane system because its lumen is in continuation with the lumen of endoplasmic reticulum. Now see what is endoplasmic reticulum. You know, endoplasmic reticulum is a network of tubular structures. It is a network of tubular structures scattered in the cytoplasm. So, endoplasmic reticulum divides the cytoplasm into two. They divide intercellular spaces into two compartments. That is luminal, means inside the endoplasmic reticulum, and extraluminal, means that is cytoplasm. So, endoplasmic reticulum is a network of tubular structures. This endoplasmic reticulum is composed of the following three kinds of structures, that is cisterna, tubules, and vesicles. Cisternae occurs in those cells which have synthetic roles. Cisternae seen in cells which have synthetic role. Tubules involved in the lipid and sterol synthesis, and vesicles are membrane-bound vacuolar structures. This endoplasmic reticulum is of two types. You know, there are smooth endoplasmic reticulum and rough endoplasmic reticulum. Smooth endoplasmic reticulum consists mainly tubules and vesicles. At the same time, rough endoplasmic reticulum consists of cisternae. It has ribosome attached to its cytoplasmic surfaces. The function of endoplasmic reticulum. Rough endoplasmic reticulum is actively involved in protein synthesis and secretion. You know, rough endoplasmic reticulum consists of scattered ribosome. Ribosomes are the site for protein synthesis. Rough endoplasmic reticulum so actively involved in protein synthesis and secretion. Rough endoplasmic reticulum bears enzymes for glycosylation and enzyme precursors for the formation of lysosome by Golgi complex. Remember, rough endoplasmic reticulum bears enzymes of glycosylation and enzyme precursor for the formation of lysosome by Golgi complex. Golgi complex are the part of a endomembrane system, and with the help of Golgi complex, rough endoplasmic reticulum helps in the formation of lysosome. The smooth endoplasmic reticulum is the major site for synthesis of lipids. Rough endoplasmic reticulum is the major site for synthesis of protein. Smooth endoplasmic reticulum is the major site for the synthesis of lipid. These 2 points, you must remember these two differentiating points. In animal cell lipid, like steroid hormone, are synthesized in smooth endoplasmic reticulum. Smooth endoplasmic reticulum helps in the detoxification of drugs. Have you got this point? Then we go to the next, that is Golgi apparatus or Golgi complex. Golgi bodies are present in all eukaryotic cells except in male gametes of bryophytes and pteridophytes. Mammalian RBCs sieve tube of plants, and in cells of fungi. Golgi bodies are present in all eukaryotic cells except in male gametes of bryophytes and pteridophytes. Mammalian RBCs, sieve tubes of plants, and cells of fungi. In animal cells, Golgi apparatus is either single or consists of single connected complex. In plant cell, Golgi apparatus is formed of unconnected units called dictyosomes. Remember the point? Animal cells, Golgi apparatus either single or consists of a single connected complex. In plants, it is formed of unconnected units called dictyosomes. Golgi complex is made of 4 parts: cisternae, vesicle, tubules, and vacuoles. It is made up of 4 parts: cisternae, vesicles, tubules, and vacuoles. And who observed Golgi bodies? Camillo Golgi first observed Golgi bodies in the form of a network in nerve cell of ban, owl, and cat. He observed the Golgi bodies in the form of a network. Okay. Then its main function. Its main function is packaging and storage of material. It is the storehouse. Its main function is packaging and storage of materials. Materials to be packaged in the form of vesicles from the endoplasmic reticulum fuse with the cis phase of Golgi apparatus and move towards the maturing phase. Golgi apparatus has 2 phases: cis phase and trans phase. Cis phase and trans phase. Packaging and storage materials packaged in the form of vesicles from endoplasmic reticulum fuse with the cis phase of Golgi apparatus and move towards the maturing phase. Protein synthesized by ribosome on the endoplasmic reticulum are modified in the cisternae of Golgi apparatus before they released from trans phase. Remember, the receiving phase is cis phase and releasing phase is trans phase. 2 phases: cis phase and trans phase. Acrosome formation during the spermatogenesis with the help of these Golgi vesicles. Acrosome formation during the spermatogenesis is with the help of these Golgi vesicles. And another function is glycosylation of proteins and lipids to form glycoproteins and glycolipids that are involved in the cell recognition and adhesion. Remember, glycosylation of proteins and lipids to form glycoproteins and glycolipids that are involved in the cell recognition and adhesion. The next structure is lysosome. Lysosomes are popularly known as suicidal bags because they contain the digestive enzyme acid hydrolysis. That's why they are called as suicidal bags. Lysosomes are present in animal cells except RBCs. In plants and fungi, vacuoles takes over the function of the lysosome. These membrane-bound vesicular structures formed by the process of packaging in the Golgi apparatus that we mentioned earlier. They are formed by the process of packaging in the Golgi apparatus. The isolated lysosome vesicle have been found to be very rich in almost all type of hydrolytic enzymes: lipases, proteases, carbohydrases, etc. One of the characteristics of lysosome is lysosomes shows polymorphism. What is polymorphism? Polymorphism is the existence of more than one morphological forms in a same cell. Lysosome show polymorphism, that means existence of more than one morphological forms in same cell. They are divided into following four forms: primary lysosome, secondary lysosome, residual bodies, and autophagic vacuoles. There are presented divided in the four forms: primary lysosome, secondary lysosome, residual bodies, and autophagic vacuole. And the last one is vacuole. It is the membrane-bound space found in the cytoplasm. Contains water, sap, excretory product, and other materials not useful for the cell, bounded by the single membrane called tonoplast. The membrane of vacuole is called tonoplast. The membrane of vacuole is called tonoplast, and it contains water, sap, excretory product, and other material not useful for the cell. So vacuoles are the waste management system of a cell. In plant cell, the vacuoles can occupy up to 90% of the volume of the cell. Vacuole may be of four types depending upon the content and function: sap vacuole, food vacuole, contractile vacuole, and air vacuole. What are them? Sap vacuole, food vacuole, contractile vacuole, and air vacuole. And the functions are divided according to their usage. In amoeba, the contractile vacuole is important for osmoregulation and excretion. In protist, food vacuole are formed by engulfing the food particle. The tonoplast has site for passage of number of ions and other materials into the vacuole against the concentration gradient. So it helps in maintaining osmotic pressure and turbidity. Remember this point. The tonoplast has site of passage of number of ions and other materials into the vacuole against concentration gradient. So it helps in the maintaining osmotic pressure and turbidity. Thus, we covered main points regarding endomembrane system, and in next video we will discuss some questions from endomembrane system, and this very important part. So go through the NCERT textbook and read thoroughly. Next video we will discuss some question from this part. Okay, thank you.